Let's go ahead and get started. Welcome to Immunology. I'm a little overwhelmed by how many of you there are, so we'll see how that works. This is my second time teaching this course. I'm Aaron. I'm very old-fashioned. I graduated from Bates in 1992. I know we weren't alive. Tell me that. Some of you anyway. When I was here, all professors had first names, so I'll be Aaron. If you are not comfortable with that, you can use Professor Winkler, but I might not answer to it. And I'll assume that the first name I have for you is okay, unless I hear otherwise from you. I figured out some of your names on the way in, but I just want to run, because we hit the limit and I've turned away a bunch of people, I want to make sure that everybody's here. We're missing a few people because of comp exams, some of whom will come in later. So Alex, I don't know Alex yet, right?
Yeah, he's injured, so he might, take a while to get here.
Okay.
If it's Alex.
I saw you on the first floor.
Okay. Jenna, I saw. Kiraku?
Yeah.
Yu Ying? Jenny, okay. Ryan, I saw. Brielle is here. Wilder? Wilder, got it. Caleb, I saw. Logan is here. Cody's in the comp exam. James is here. Sorry? Two James, sorry. You'll be next. Aaron Kelly is away. I'm going to slaughter yours. You pronounce it for me. Kai? Okay. Tra is in comps. Emily Ladd's in comps. Rebecca. Hi. Rachel. Hi. Alina's away. Colin is here. Ilana is here. James, the other James is here. Sarah. Hi. Jacqueline. Jackie. Marissa is here. Courtney is here. Hi. Regan or Reagan? Reagan, hi. Ian is here. Chloe is here. Nabil is here. Nancy. Oh, Nancy. Hi.
Hi.
And Merritt?
Marie.
You're Alex.
Alex, yeah. Thank you.
So now that Nancy is here, we can get started. This is a new format for class. Until this year, it's been a lab class. It's been 2 days a week with an afternoon lab. Because I'm traveling from Boston, we collapsed it to 1 day a week, and there's no lab. So you're not really positive on your perspective. But it's going to be a little bit intense, I think, having 2 lectures on Friday afternoon when everybody wants to go to sleep or go away or do something else. So we'll do our best to make it interesting. Those of you who have read the syllabus will see that there's a number of texts offered. I had to change the text because this one, TAC, went out of print last year and they're not releasing the new one until next year. I will not be teaching directly out of any of these books. Most of the material that I give you will be what I've gathered from other sources. I think Param is a good source. It's a bit expensive, but I think it's the best possible source, but it's not the gospel. What I'm going to try to do is teach you a way to think and learn immunology, not a bunch of stuff to memorize. So there won't be any in-class tests, there won't be any multiple choice. So you are all scientists of some sort or another. So when you leave Bates, no one is going to unless you go to medical school or graduate school, nobody's gonna give you memorization tests. You're gonna have the whole world of knowledge to learn from, and I hope that that's what you do in here. So what we're gonna try to do is give you the basic vocabulary and understanding of immunology, and together we'll try to learn new ideas and come up with interesting things to think about. Those of you who have looked at the syllabus will see that after the second class, we're going to start having discussions, and these are going to be student-led. It's going to be a little bit harder than in the past because there's so many of you. Hopefully we can divide into about 7 groups of 5. Previously we've done that with lab groups, but since we don't have a lab, I'll do it kind of randomly. So the idea there is that you can bring some new idea to the class in one of the general areas we're discussing, and give a short introduction, stimulate some conversation. So that leads into evaluation. Previously, I've given a quiz every other class. It's not really a grading tool, it's just to make sure people are keeping up, and it helps identify people who are having an issue. There won't be very many. I think it'll just be can't remember how many are on the syllabus, maybe 3 or 4. You shouldn't have to study for it. If you are coming and reading, it shouldn't be an issue. We may even get rid of them if they're not helping. The important part of the grading will be the take-home exams. As I said, I'll give you a choice of questions. You won't have to answer every one, probably. You can pick a couple to answer. You can use any material from the class or the internet or other textbooks, as long as it's fairly validated to answer the question. But because there are so many of you, it's going to be valuable to keep them as brief as possible. You could all write a thesis in a week probably, but we're going to try to keep it on the 1 or 2 concise paragraphs. So you shouldn't spend the entire week doing your exam, a couple of days. And what you're going to be graded on is integrating what we've learned and answering a question that either hasn't been answered or will never be answered. Just show me how you can synthesize knowledge. The only problem with that is that's subjective grading. So we'll use the grading process as a dialogue to make sure that we're all learning together. I already talked about discussions. If you see any, the breakdown down here is so there's a huge portion of participation, either in class or in discussion. So we all learn in different ways. So if you're a talker, more of a writer, do more talking. I reserve the right to move us on if it's not on topic, but I encourage you to interrupt any time. If you see any problems with the syllabus in terms of dates or conflicts or whatever, just let me know and I'll change it.
I just have a question about the quizzes. Are they going to be on the readings that were like assigned prior to it?
Yes, they will not be on future knowledge. They'll be on
So it's
whatever
not,
we've
so
covered.
we've talked about it in class already?
Yes.
Okay.
And really big picture stuff, just to make sure that somebody's not on another planet. Anything else? And if you guys have ideas, I mean, I've never taught for 3 hours at a time, so we're gonna have to play this by ear and see how it goes. If you have ideas about how we can organize our weeks to learn more effectively, I'm open to those. Right now, my plan is to basically give 2 lectures every Friday. We'll put the discussion either between them or after the second one. We'll have a break in between, approximately halfway through. You're grown-ups, so wander in and out for food, water, entertainment. toilet, whatever you need to do. But when you're in here, you can keep the texting and other distractions aside, and you don't need my permission to miss class, but if you need help keeping up, just let me know. I'm not going to be taking attendance after today. So any other administrative stuff? So anyway, a little bit, I started on my background. I came to Bates in 1988. I had known for a long time that I was interested in immunology, so as soon as I got here as a freshman, I petitioned to take this class, and they let me in, which was probably a bad idea, so I was totally lost. But at that time, Carnegie was just this half of the building. It was expanded to its current size while I was here, and the professor of immunology was named Blake Whitaker. He actually went on to start a company on Main Street in Lewiston called Immunet. It lasted 5 years, and I was the second employee. That's what happens to most startups, especially in Lewiston, Maine. But during that time, my predecessor, Pam Baker, came and became an immunology professor, and she was my thesis advisor while I was here. And she went on to become the dean of the faculty a few years ago. She retired last year. But I taught this course 2 years ago because when she was promoted to dean of the faculty, she asked me to take the class. So I'm just an adjunct professor. I'm a scientist in the inflammation and immunology department at Pfizer in Boston. I've been to med school, I've been to grad school, so when I taught this course previously, there are a number of people who were trying to make decisions about work, graduate school, med school, etc. I'm happy to meet anytime on Fridays that anyone would like to discuss that, immunology, anything else. Anytime after about 10 in the morning, so that's about as early as I can get here, or after class. So keep that in mind. That's about it. So I gave you the detail of the syllabus. We'll do a little philosophy. So those of you who haven't printed out the syllabus, the truth is a beautiful and terrible thing, and should therefore be treated with great caution. However, I shall answer your questions unless I have a very good reason not to, in which case I beg you to forgive me. I shall not, of course, lie. The great wisdom of Albus Dumbledore. I think actually I won't tell you any truths in here because science is just the best description as we understand it at the moment. Nothing is set in stone. So I will answer any of your questions and the information I present with the best current knowledge as I can synthesize it. You will ask a lot of questions that I can't answer, and I'll try to tell you truthfully when that's the case. And some of them won't have answers, and some of them you'll answer in the future in your research or careers. But I will help you to find the answer as I can. And if you discover that what I've told you is not true, I hope you'll point it out to all of us. Okay, so one of the first immunologists, and I think it's an interesting story, which is why it's in the first chapter of most immunology books, was Jenner. And his basic observation was, at a time when many people died of smallpox, that milkmaids didn't get smallpox. And milkmaids, however, because they were in close contact with cows every day, did get a disease called cowpox, which looked like smallpox but wasn't fatal and was a much milder form. So he took this observation that milkmaids that got cowpox never got smallpox and formed a hypothesis. There's something about getting cowpox that keeps you from getting smallpox. So what is it? What is it in that process? So this was long before the germ theory or any idea about microbiology. But what he did was he took a syringe and took the vesicle contents of a milkmaid with cowpox and used it to inoculate a child, James Fitz, and then waited 2 months and injected smallpox, or actually the pus from a smallpox wound into James Fitz. Does anybody see a problem with that? Yeah, I guess it's obvious there's some ethical issues there. But then, even at that time in the 16th century, there was a medical literature, and so he wrote up his experiment to describe his observations that cowpox was not only transferable but could prevent smallpox. So obviously we can critique the ethics in terms of infecting a patient with disease. Not to mention the size of the sample. It was an N of 1 experiment. There was no purity in terms of disease he transferred. He knew nothing about what he was transferring from the milkmaid to James. And there were no controls, obviously. There was nobody who received a placebo injection and then smallpox. Okay. So immunity came from approximately the same time of the great pandemics of flu and smallpox. It was because people noted that there were those who did not get those pandemics, and so immunology was basically the study of those who did not get those pandemics. It was actually about 100 years later that James Koch initiated the germ theory of disease, basically describing this process of transferring disease from one to another. And even though the actual germs themselves could not be visualized at the time, he set up a set of rules to describe how infectious disease works. And I just, 1 second on an aside, your textbook focuses a lot on infectious diseases, viruses and bacteria that cause disease. You have good virology and bacteriology classes. We're not going to focus on the organisms at all, except to use them as examples of immunology. So, with a few notable exceptions like HIV, because it's immunosuppressive, we're not going to talk about the pathophysiology of the organisms themselves. So, as a general rule, you can ignore the portions of the book that go into great detail about what different bugs cause. We'll just use them as examples. Okay, so Koch's postulates were that the microorganism must be found in abundance in all organisms suffering from the disease, but should not be found in healthy organisms. So, obviously, the example is the milkmaids who had cowpox all had whatever that organism was. You could then, the real step forward then was generating cultures. So you must be able to isolate from that pus the actual organism that caused the disease. And this was obviously easier originally with bacteria than with viruses, because viruses require growth in living cells. And then, of course, you had to take that pure microorganism and transfer it to a healthy organism. You couldn't just take the pus from the cowpox milkmaid and inject somebody. It had to be the pure organism. And then when re-inoculated, it had to generate the same disease, so it had to be transferable. So this is not necessarily an important tenet of the course, but it helps you keep in mind what a novel idea it was at the time that there were living organisms that caused disease. So prevention of infectious disease is just one of the important jobs of the immune response. So at a basic level, it distinguishes self from non-self. However, it minimizes all kinds of threats. Those from self, like cancer. Those from non-self, like inhaled particles. So your immune system is one of the systems required for clearing particles that you breathe in. And then there are non-self infectious, like bacteria, fungus, and virus. And there's a very complex interaction between those microorganisms and the immune system. In fact, you probably already know that about 2 pounds of each of us is the microorganisms that inhabit our digestive tract, and we're not infected with those microorganisms. They don't cause disease, so, but they are kept organized by the immune system and kept in the right spot. And then, of course, the basic traditional understanding that the immune system counteracts infection. And infection is defined by attachment to, entry, and/or expansion of pathogens in the host. And this could happen outside of our cells or inside of our cells. Okay, so for the rest of this first lecture, we'll just go over some basic concepts. Everybody good so far? Okay. So immunology is mediated not just by cells, but also by soluble factors. So we're generally divided into cell-mediated, which is predominantly white blood cells, leukocytes, versus humoral, which is soluble factors, which include antibodies, cytokines, and complement. And then a very basic concept in immunology is dividing the innate from the adaptive immune system. So innate immunity is genetically encoded and broadly specific and older evolutionarily. So in other words, there are more evolutionarily ancient animals that have innate immune systems that don't have adaptive immune systems. The adaptive immune systems, as the name implies, are acquired within an individual organism. So each one of your adaptive immune systems is totally different because each one of you has a different genetic background. You have been exposed to different environments. You've had different infections. So you each have your own unique adaptive immune system, but for the most part, you have the same innate immune system with the normal variability of the human genome. Okay, so innate immunity extends beyond cells that we think of as the immune system, like white blood cells. So it involves barriers as well. So there are non-specific parts of the innate immune system, such as skin and gut epithelium. So those are the anatomic and physiological barriers. So basically the easiest way to not be infected is to not have a break in your epithelium. I think most of us have had a scratch or a cut, and Mom always taught us to wash it out with soap and put a Band-Aid on it. And those of us who didn't do that ended up with redness and swelling because it was infected. Then the specific aspects of innate immunity, there are actual specific reactions to those infectious microorganisms. And we have in the innate immune system, we'll get into in a moment, receptors that identify broad classes of molecules that are associated with bacteria, viruses. So there are particular common factors in the coats of bacteria like LPS, or specific nucleic acid sequences like double-stranded RNA that we don't make, that viruses make. And so those broad classes, which we'll discuss in the next class. So that's a specific immune response. However, we all have the same receptor for those specific immune responses. So those receptors are called pattern recognition receptors. And there's no memory. The first time you respond to bacteria with that pattern recognition receptor is exactly the same as the 10th time, the 100th time. There's no memory, there's no change in the immune response. So one important class of those pattern recognition receptors is called toll. There's at least 10 known tolls, and we'll go into those in detail in our innate immune lecture next Friday. This was actually first identified in a knockout experiment in Drosophila. So this is a fruit fly. If you've taken, I don't know, maybe you don't do it anymore. When I was here in genetics, we did fruit fly experiments to see how you get white eyes or red eyes and crumpled wings or flat wings. But if you take a Drosophila and knock out a molecule that was called toll, then it dies, and the reason it dies is because of fungal infection. So all these hairs are not a normal part of Drosophila structure. These are all the fungal spores growing on the Drosophila. So if you knock out toll, all the Drosophila die very quickly, whereas the normal ones survive. So this was the first identification in the fly of these pattern recognition receptors. And so you can see this happened less than 20 years ago in 1996, and after I took immunology here. So this is a whole realm of immunity that didn't exist 20 years ago. Okay, so that's just an example. There'll be much more detail later. The tools that innate immunity uses, so we talked about the receptors. They are broadly specific to general categories of molecules that are non-self. The tools that the innate immune system can use to respond are pretty basic. They are phagocytosis, in other words, taking up those materials that are identified as non-self. There's killing, basically using basic defense mechanisms to kill those invading cells. This is especially true with regard to bacteria, because those could be subjected to oxidative burst and pore formation. And then there's an inflammatory response, in other words, changing the local tissue environment to begin an immune reaction. And so just because I think it's cool, I'm going to show you what
Neutrophils
phagocytosis
are white blood
looks like.
cells that hunt and kill bacteria. In this spread, a neutrophil is seen in the midst of red blood cells. Staphylococcus aureus bacteria have been added. The small clump of bacteria release a chemoattractant that is sensed by the neutrophil. The neutrophil becomes polarized and starts chasing the bacteria. The bacteria, bounced around by thermal energy, move in a random path, seeming to avoid their predator. Eventually, the neutrophil catches up with the bacteria and engulfs them by phagocytosis.
So that's a bit silly, but the concept is that neutrophil has no adaptive immune tools. All it has is pattern recognition receptors. So it is identifying components that are coming off of the bacteria. No, you don't need to see. Oh. You can find really gross stuff on YouTube. Yeah, just type in pus and see what you find. Anyway, so there are molecules coming off of that bacteria that are sensed by the pattern recognition receptors. So they're the same that any of us have. If you put any of our neutrophils in a tissue culture dish, they would have the same basic response. And they would basically chase down the bacteria and engulf it if possible, or kill it extracellularly if not. So in terms of the organism beyond an individual cell, likewise, before there was microscopy, before there was the germ theory of disease, medicine had described inflammation. And there are five cardinal signs. I'll spare you the Latin, but basically redness, swelling, heat, pain, and loss of function. And those are all characterized by this ingrown toenail here. Basically, the redness has to do because there's vasodilation, so there's increased blood circulation there. There's swelling because the circulatory system has become leaky, so there are fluids and cell leaving the circulatory system. There's heat because there's not only increased circulation, but release of local factors that cause heat. There's pain because the immune system is releasing substances that notify the neural system that there's local damage. And if there's enough inflammation, there's loss of function. Okay. So that's just a general high-level tour of innate immunity. We'll spend pretty much all Friday next week getting into it more specifically. Any questions at this point? Nobody's asleep yet. Wilder? Nobody's asleep, right? Okay. Okay, so adaptive immunity. In contrast to innate immunity, there's specificity. Like I said, we each have our own set of adaptive immune systems, and we're going to spend maybe close to half the class throughout the semester talking about the mechanisms of adaptive immunity. There's some, I think, really unique molecular biology that really is seen nowhere else in the body in terms of how immune cells actually edit their genetic material in order to respond to threats. And that's how each of us has our own adaptive immune system, because each T cell and B cell has the ability to edit its genetic material so that it only responds to a very specific single molecular pattern. So, as I said, the receptors, unlike the pattern recognition receptors, which recognize one single broad class of a shape, and it's the same for all of us, they can, basically there's infinite diversity. Anything that you can code with nucleic acid can be made into a receptor, and they're essentially genetically encoded molecular shapes. The most important cells of the adaptive immune system are T cells, and that unique receptor on T cells that identifies shapes is the T cell receptor, whereas B cells express antibodies either on the surface or they secrete them. And the important feature is memory. So this is why, if we've been vaccinated to the right flu, we create a memory against influenza and then don't get full-blown flu. And why we shouldn't, we'll talk about specific situations of loss of immunity, but in general, those of us who grew up before the chickenpox vaccine all had chickenpox once in childhood and never had it again. And hopefully you guys who've grown up getting the chickenpox vaccine will never experience it. Okay, so some special features. While innate immunity, there's no specific mechanisms for intracellular interaction. Adaptive immunity has very specialized cell types. So in the example we gave where we showed a neutrophil, every neutrophil is the same, every neutrophil does the same thing, they don't try to work together. Whereas in the adaptive immune system, T cells and B cells come in a variety of flavors and they work together to coordinate the immune response. Again, just to repeat important points that can come up in quizzes, there's memory in adaptive immunity. So the same response to the same agent evokes a faster, more effective, and more specific immune response the second time. So effective that you might not know that you got an infection the second time. And then there's tolerance. You can think about the incredible danger involved in having cells that could respond to any molecular shape in the universe. That means they're fully capable of responding to every shape your own body makes. So in order to have an immune system that can get rid of any threat imaginable and yet not respond to all the molecules your own body makes is a very fine balance and requires an active process of tolerance. So this summarizes, it's a nice table from Janeway. Your book, actually the Parham book, is a reduction of Janeway. You can also use Janeway if you want, it's just a much bigger volume. But basically it highlights the differences between innate and adaptive immunity. So secondary response is always the same in the immune system. The receptors are fixed, they aren't reorganized. Response is not clonal, respond to observed molecular patterns. And there's conservation evolutionarily. The adaptive immune system has memory, secondary responses are changed, there's genetic reorganization involved in that, and there is imperfect discrimination between self and non-self, which requires tolerance. And there are some overlapping and distinct cytokines that we'll talk about in other classes that are key in communicating between the cells. Any questions on this? Okay. So, as I've implied so far, the immune system is incredibly powerful and incredibly necessary. There are a variety of inherited immunosuppressive diseases, and if you're born with one of those diseases, kids don't tend to live very long. We'll talk about some of those in later classes, but it's a system that's necessary for life, and it's an incredibly well-balanced system. With too little immunity, we can be plagued by infection, cancer, and dead tissues and unhealed wounds. And you can think about that. I think many of us have learned a little bit about HIV, so a little bit of a defect in one of the cells of the immune system, you get susceptible to all of these situations. In the case of too much immunity, we are subject to hypersensitivity, allergy, and autoimmune diseases such as rheumatoid arthritis and lupus, MS, etc., all of which we'll discuss this semester. This is probably too small to see clearly, but and it's not for memorization. Charts like this are available in every textbook, and online. You should always be able to reference them. But it just brings about the point that the cells of both the innate immune system and the adaptive immune system, in terms of leukocytes, all derive from a single hematopoietic progenitor cell that lives in the bone marrow and reproduces. And then there's an incredibly complex system of growth factors that then causes successive differentiation of that hematopoietic progenitor cell into every cell of the innate and adaptive immune system. So the first differentiation is from that hematopoietic progenitor cell to a common myeloid progenitor, which leads to all of the innate immune system, including platelets and red blood cells, so cells outside of the immune system actually. And then the common lymphoid progenitor, which gives rise to T cells, B cells, and NK cells. So all from one individual cell, but the earliest step in their differentiation. And then the earlier steps in these, not every mature cell comes from a straightforward differentiation from a hematopoietic progenitor. These earlier stages, in terms of the committed progenitor cells, can also reproduce. So there's also a renewal of later stage progenitor cells as well. So the location of those hematopoietic events varies during gestation. In the earliest stage in the yolk sac, there's already hematopoiesis going on, and blood actually exists during the first trimester. So there's obviously hematopoiesis going on. That hematopoiesis quickly transitions into the fetal liver, where most of hematopoiesis goes on during gestation, and then later in the third trimester, the hematopoietic cells relocate from the liver to the bone marrow. And in all of us today, hematopoiesis goes on in long bones. And so there are actually cases where fetal transplant of hematopoietic cells has been done intrauterine, so that a genetic defect is identified during gestation, and hematopoietic stem cells have been transferred into the fetus during gestation, so that they actually are born with a functional immune system. Okay, so to round out this first lecture, we'll just discuss some specifics of the individual cell types that I've alluded to so far. So, as I mentioned, one of the most numerous and important cells of the innate immune system are neutrophils, the ones we saw on the film. They're the earliest and most numerous cells in an immune response. They have pattern recognition receptors and immunoglobulin receptors. This means that they can bind to antibodies. So if you have antibodies in your circulation to a particular kind of bacteria, those bind to that bacteria when it gets into your skin. The neutrophil, even though it's of the innate immune system, it's able to identify something foreign has been coated with antibody and respond to that. So as we saw, they can respond by phagocytosis and then intracellular killing, or for larger pathogens, they can perform extracellular killing. And there's a bystander effect, so not only will they kill target cells, but they'll also kill cells if necessary. And that's done with an oxidative burst, which we'll describe in a minute. So the results of the neutrophil response is death. The pathogens, the neutrophils themselves seldom survive their response to a pathogen, as well as I said, bystander cells. And they release a host of potent inflammatory mediators to recruit other innate immune cells and adaptive immune cells to the site of inflammation. So, Kubi is also another good source if you're looking for different textbooks of immunology. So, neutrophils, eosinophils, and basophils are the three granulocyte cell types. Neutrophils are the most common white blood cells in the circulation, while eosinophils and basophils are incredibly rare, less than 0.1% of your circulating cells. Neutrophils have a specific set of growth factors that cause their differentiation, and as I said, they're the most common white blood cell and are responsible for early immune responses. Eosinophils and basophils have important roles in responses to extracellular pathogens, parasites, etc. And they mediate a lot of the pathophysiology of allergic disease.
What do you mean by granulocyte?
Okay, good question. The cells of the circulatory system were named originally because of how they appear under the microscope, and the basic staining of white blood cells in a smear is with hematoxylin and eosin. And these cells appear very granular. They're essentially packed full of vesicle-defined granules. And so maybe I'll bring some images next time, but you can hardly see the nucleus because they're so packed full of granules. And that's because when they undergo an inflammatory response, they release all those vesicles. They release cytokines and enzymes and free radicals, basically, to kill anything around. So they're packed full of these vesicles, whereas other white blood cells, as we'll see in a minute, don't have that appearance of a granular, they don't have a granular appearance because they're not full of vesicles. Okay, so the other important circulating immune cells are monocytes. They're about 10% of white blood cells. They have a lower frequency than the neutrophils, and they don't have that granular pattern. Again, they have the same or similar pattern recognition receptors and immunoglobulin receptors. They're important for phagocytosis and intracellular killing, and they make a lot of cytokines. And as we'll see later in the course, they actually differentiate into antigen-presenting cells. So they process molecules to present them in a form to the adaptive immune system that can be recognized by the adaptive immune system. So the result of the monocyte response is death of pathogens, inflammatory mediator release, and then antigen-presenting cell differentiation in the tissues. So in circulation, the monocytes have an ample cytoplasm. They have this characteristic kidney-shaped nucleus. In tissues, they acquire different shapes with a lot of appendages that are adapted for living in their particular environment. And again, in every tissue, whether it's skin, digestive tract, liver, there's a specific brain, there's a specific type of macrophage that is differentiated in the tissue from circulating monocytes. So again, there's the common myeloid progenitor in the bone marrow. It releases neutrophils and monocytes into circulation. Neutrophils essentially stay in the circulation or undergo extravasation for an inflammatory response, whereas monocytes can actually gain new specificity in specific tissues. They can be recruited to an active site of inflammation from the circulation, or they can become resident cells in specific tissues. In the skin, those are Langerhans cells. In the liver, they're Kupffer cells. In their bone, they're osteoclasts, which are key for bone remodeling and growth. And in the neural system, they're called microglia. Similarly, there are specific types of dendritic cells that differentiate in tissues. The specific names of macrophages and dendritic cells is not important. It's just important to know that all of the important antigen-presenting cells differentiate from monocytes or the common myeloid progenitor. Whereas macrophages are good at doing a lot of phagocytosis and taking up particles and inflammatory responses, dendritic cells are really the professional antigen-presenting cells. So they're predominantly responsible for sampling the antigens that enter our body, either through the skin, lung, digestive tract. They're constantly capturing substances from those tissues and migrating to lymph nodes, the structure of which we'll describe later, and presenting that antigen to T cells and B cells. And so in those lymphoid structures, there are cells of the adaptive immune system constantly saying, not a problem, not a problem, not a problem. Wait, that's a problem. So it's the cells of the professional antigen-presenting cells, the dendritic cells, that are responsible for taking substances from the tissues to the innate immune system, to the adaptive immune system. Okay, so the last important class we're going to talk about of white blood cells are the lymphocytes. So again, this predominantly makes up the adaptive immune system. They're responsible for memory of pathogen-specific immunity. As we mentioned, their specific receptors are the TCR, the T cell receptor, and the B cell receptor. They also have pattern recognition receptors and immunoglobulin receptors like the innate immune system. T cells are responsible for mediating the immune response, direct cell killing, promoting B cell responses, and cytokine production, which controls the characteristics of the immune response. The B cells are predominantly responsible for antibody production, but they can also present antigens, just like dendritic cells can. So the result of the immune response with adaptive immune cells, again, is a targeted immune response that's not only specific to the pathogen, but also has a specific characteristic in terms of if you have a viral infection, that is one kind of immune response. If you have a parasite infection, that's another kind of immune response. If you have a bacterial infection, that's a different kind of immune response. So the adaptive immune system controls not only the specificity, but the characteristics of the adaptive immune response.
So do the lymphocytes release the T cells, or are they the T cells?
So lymphocytes is a big class of white blood cells that comprise the adaptive immune system. T cells and B cells are types of lymphocytes. And then within those, we'll have 3 or 4 lectures each on B cells and T cells, and there are subtypes of B cells and T cells as well.
For B cells that present antigens, can those antigens be broken down by neutrophils and T cells, or just T cells?
Okay, so the way that B cells present antigen is different in the way that dendritic cells present antigen. Because B cells, even the ones that are antigen presenting cells, express a specific B cell receptor. So when a B cell, we'll come back to this, so don't worry if we get lost. When a B cell responds to a threat, it's because the B cell receptor on the surface of that B cell is specific for that threat, for a particular viral or bacterial molecule. And so it already has specificity for that molecule. It already has an antibody on its surface. And so it ingests that, whatever's attached to that antigen, digests it and presents antigen on the surface. And it can present antigen on the surface to T cells or B cells. And each of those T cells and B cells has its own set of specific molecules on the surface that may or may not recognize some component of that bacteria. So it basically, antigen presentation is just digesting anything that's either self or non-self and presenting little pieces of it on the surface so that it can be sampled by the immune system. Most of what is presented will be self or non-threat, and there'll be no response. A very small percentage of that will be non-self and will be recognized by a T cell receptor or a B cell receptor. And so when those molecules are recognized by those T cell and B cell receptors, then those T cells and B cells respond and clonally expand. And that's when you get an adaptive immune response. So neutrophils are only involved in the sense that they usually arrive first at a site of if you break a blood vessel or you cut yourself, they'll automatically leave the circulation and enter the wound space. If it's a sterile wound, they won't do much. They'll just die there. If there's a foreign molecule that one of their pattern recognition receptors recognizes, they will release cytokines that recruits more cells. They'll have an oxidative burst to try to kill that cell. So the neutrophil is really pretty much separate from the adaptive immune response. Whereas those monocyte-derived cells, the dendritic cells and the macrophages, they bridge the innate immune response to the adaptive immune response by presenting antigen. And we'll have a whole class on antigen presentation. And the reason you need antigen presentation is because the T cell receptor and the B cell receptor only work in very specific contexts. They have to see non-self in a very specific way. And that's one of the ways that the body limits the immune response, because the immune response is so powerful it could easily be fatal. You think about people who have terrible allergies have anaphylactic responses. So one of the ways that the immune response is limited is by making very specific ways in which B cell receptors and T cell receptors have to be activated. So it can't just see non-self, it has to see non-self in the context of the right receptors on an antigen-presenting cell. Did that answer the question?
Mm-hmm.
Okay, so as was asked, that there are three subsets of lymphocytes: B cells, T cells, and NK cells, so-called natural killer cells, and we'll have classes on each of these and their specific function. And again, they have their own overlapping sets of hematopoietic factors and subsets. Okay. So, one more little brain break. Michael Savage is a conservative talk show host. And when I was looking for demonstrations of free radicals, I was surprised to find out that he had important information to share.
1-800-449-8255, michaelsavage.com. I don't want to talk about the news, I really don't. I would rather talk about phagocytosis. Those of you who understand what I just said, it is a medical term. Phagocytosis is a medical term that I recall from my days of studying about free radical pathology and antioxidants. And phagocytosis, you can't even say words today, they're in the medical books for fear somebody's liable to complain to somebody. I do have a doctorate in epidemiology and human nutrition. I do remember my studies of antioxidants. Everyone says antioxidant, they don't know what it means. Oh yes, I'm taking my antioxidants. You don't know what it means, but you're taking them. It's like saying I'm eating my carrots. You're not a rabbit. Now it is true that you should eat carrots once in a while and your broccoli. But how many of you really know what a free radical is? You have no idea what it is. Harry Reid is a free radical. Nancy Pelosi is a free radical. Andrea Mitchell is a free radical. Charlie Rangel is a free radical. Barack Obama is #1 free radical. Problem is no one's scavenged any of them yet. That's why I'm talking about free radical pathology. They're out of control. They are devouring the body politic. We absolutely need antioxidants to control them. See, I can put this into a medical context, and we might actually get somewhere. You're talking about a degenerative disease. What they're doing to the body politic is like a degenerative disease that's about to kill a patient. So if I told you that I've written about the free radical theory of aging in 1972 in a popular book called Nutrition Against Aging, you'd say, but sure, he didn't.
That's all I can handle. Those are not my political views. And really it just highlights the fact that there's all kinds of useless information on the internet. So beware what you learn out there, even from people who say they have a relevant degree. So just to let's see what we have left. Okay, so a couple more slides. So we described the tools of the innate immune system is basically phagocytosis and oxidative burst. And the oxidative burst can happen intracellularly or extracellularly. In this case, it shows the process of a receptor of any sort, either one of the innate or the adaptive immune system, identifying an extracellular bacteria, phagocytosing it into a membrane-bound vesicle, and then using a variety of mechanisms to kill enzymes and free radicals. So free radicals, you've all taken some sort of chemistry, are basically unpaired, are these ions with these submolecular particles with free radicals, unpaired electrons in them, and those are very reactive, so they react with molecules of the pathogen and destroy it. So although there's a lot of literature about the importance of eating free radical scavengers, they're an essential part of the immune response as well. So there's an important class of immune diseases called chronic granulomatous disease, and they result from basically mutations in any one of the components of the multi-protein enzyme complex that produces free radicals. So the percentages listed here are some of the important inherited diseases. None of this is important to memorize. It just points out the fact that the oxidative burst of the innate immune system is incredibly important for fighting infection. And if you do have one of those mutations, you essentially have no ability to kill extracellular microbes. And so the body's only solution is to wall them off, to create these granulomas, which is basically just a walling off of the tissue with macrophages and structural cells because the bacteria can't be killed. And with somebody with chronic granulomatous disease basically ends up covered with these granulomas of infections that can't be cleared but are simply walled off. Okay, so let's take a break for 10 minutes or so. Come back and do the next lecture. important points from this first lecture are simply the difference between an innate and adaptive immune system and how important regulation is. Too warm? I don't know.
It's always warm.
Ian, how are you?
Not too bad.
Printing in the second lecture right there. I assume it was like lecture one. That
I see, no.
no. I'm just different block right 10 minutes.
Oh, okay.
It's actually blocked.
Imagine that the granule lattice disease. So how do you treat that? Could you just lice the, I don't know, walled off things
So
or?
they get a lot of antibiotics, bacteria, and then it's basically as you're applying, there's a lot of effective surgery.
Yes,
So a lot of those people function.
So you just take a, like a lifelong course of antibiotics essentially?
Throw down the bell.
What?
I should advertise.
No,
I
there-
got
it's obviously a
sugar-free.
big opportunity for gene therapy.
Yeah.
If you could get rid back a functional gene, somatic stem cells, that's been done.
Yeah. No, it's just,
It's metal. Maybe.
Are you gonna be cold?
Yeah, it could be actually.
I guess, yeah. I know you don't have to. No,
class.
It's me. I haven't seen you at all.
Yeah, well, I just got back yesterday.
Oh, okay, that's fine.
So morning, it was so great. This is like, oh yes, I like this, my house, I turned the heat up as high as it can possibly go, and it's so cold in there. I
Alex, you unsuccessfully put his leg under yourself.
Yeah, I did.
And that's what caused that.
Yeah, I fell on my leg.
Putting you off season.
Pretty unfortunate, to be entirely honest, but I sort of accepted it. I mean, I sort of accepted it. I kind of have to accept it.
I mean, yeah, I mean, it's not like you can be like, oh yeah, I'm gonna get better.
I don't think
Yeah.
there are like that much
It'll be like
sports
2
too.
months probably at least till I'm running.
It's just like new people.
I mean, there's nothing I can do now except let it do.
I mean, I watched. Oh,
Yeah,
I watched the first Lord of the Rings.
But then that means you could probably do like track under.
Yeah, maybe
So
like
have
strength
watched-
track. I'll probably start swimming. Something like that.
No, I watched one.
Yeah, it's like strength
Where
training.
they do like racing.
If I want to rerun it for a while.
Hey.
I'm really just
Not to like